Sunscreen that includes zinc oxide, a common ingredient, loses much of its effectiveness and becomes toxic after 2 hours of exposure to ultraviolet radiation, according to a collaboration that included Oregon State University scientists. The toxicity analysis involved zebrafish, which share a remarkable similarity to humans at the molecular, genetic, and cellular levels, meaning many zebrafish studies are immediately relevant to people. Findings were published in the journal Photochemical and Photobiological Sciences. The research team sought to answer important but largely neglected questions regarding the massive global sunscreen market, predicted by market data firm Statista to be worth more than $24 billion by the end of the decade. The questions: how stable, safe, and effective are sunscreen ingredients in combination rather than as individual compounds, which is how they are considered for Food and Drug Administration approval. And what about the safety of any chemical products that result from reactions caused by exposure to sunlight? The researchers say that sunscreens are important consumer products that help to reduce UV exposures and thus skin cancer. But we do not know if the use of some sunscreen formulations may have unintended toxicity because of interactions between some ingredients and UV light. What the public thinks about sunscreen safety has caused manufacturers, often based on limited data, to use lots of some ingredients while limiting others. For example, oxybenzone has effectively been discontinued because of concerns that it harms coral reefs. And sunscreens containing inorganic compounds like zinc oxide or titanium dioxide that block UV rays are being marketed more and more heavily as safe alternatives to the organic small molecule compounds that absorb the rays. Scientists made 5 mixes containing the UV filters, the active ingredients in sunscreens, from different products available in the United States and Europe. They also made additional mixes with the same ingredients plus zinc oxide at the lower end of the commercially recommended amount. The researchers then exposed the mixes to ultraviolet radiation for 2 hours and used spectroscopy to check their photostability. That is, what did sunlight do to the compounds in the mixes and their UV protective capabilities. The scientists also looked at whether the UV radiation had caused any of the mixes to become toxic to zebrafish, a widely used model organism that goes from egg to swimming in 5 days, and found that the UV exposed mixture without zinc oxide did not cause any significant changes in the fish. But scientists saw big differences in photostability and phototoxicity when zinc oxide particles were added, either nanoparticles or the larger microparticles. With either size of particles, zinc oxide degraded the organic mixture and caused a greater than 80% loss in organic filter protection against ultraviolet A rays, which make up 95% of the UV radiation that reaches the earth. The researchers say that the zinc oxide induced photodegradation products caused significant increases in defects to the zebrafish they used to test toxicity. That suggests zinc oxide particles are leading to degradants whose introduction to aquatic ecosystems is environmentally hazardous. The researchers say that the findings would surprise many consumers who are misled by nano-free labels on mineral-based sunscreens that imply the sunscreens are safe just because they don't contain those smaller particles. Any size of metal oxide particles can have reactive surface sites, whether it is less than 100 nanometers or not. More important than size is the metal identity, its crystal structure, and any surface coatings.